Hello everybody, welcome back to Online Learning with Miss E. This is the Central Dogma of Molecular Biology Part 2. So if you missed Part 1, go back, watch that video first. But today is Part 2. We are talking about transcription. And here's a picture of a super cute hedgehog to help you power through this lecture. And if you wait till the end of the video, I have even cuter pictures of hedgehogs, so watch until the end. So first, let's think back to video part 1. The last video, we reviewed DNA and talked about how it carries the instructions for life, how important it is for our cells. Now, if DNA is the code for life, how does that message get to other parts of the cell to control all cellular functions? This is where we have our central dogma. Remember, our central dogma says that we go from DNA to RNA, and then from RNA to protein, going from the instructions of life to the actual expression of the proteins that we need. So today, we're just talking about the first step in this process. How do we go from DNA to RNA? So, from DNA to RNA, we know that DNA holds all the instructions for life. However, DNA is stuck in the nucleus. It can't leave. So if it can't leave the nucleus, how can DNA provide directions to the rest of the cell outside of the nucleus to perform all the necessary cellular functions? Well, DNA needs a messenger to deliver directions outside of the nucleus. And this messenger is going to be RNA. So we can see in this picture here, we have our nucleus right here, and our DNA is stuck inside. It's not going anywhere. But what it can do is DNA can be transcribed or copied into this other thing called mRNA, and that RNA is able to actually get out of the nucleus into the cytoplasm, where it can ultimately direct the production of proteins. So our mRNA, our messenger, is going to take the information from DNA, shuttle it outside of the nucleus into the cytoplasm, where we can make all the proteins that we need. So right now, we're just going to talk about this first process here, going from DNA to RNA. All right? So, let's take a closer look at RNA. What is RNA? RNA is ribonucleic acid, which sounds very similar to DNA or deoxyribonucleic acid. Both are nucleic acids, meaning that they are composed of a sugar, a phosphate, and a base. However, in RNA, the sugar, the 5-carbon sugar, is called ribose. And in DNA, the 5-carbon sugar is deoxyribose. And there's a very common sense reason why they have these two very similar names. Let's look here at the bottom of the ribose sugar. You can see here that ribose has 2 oxygens. 1, 2. In deoxyribose, we are missing an oxygen. Ribose has 2, oxygen just has the 1. So it got its name deoxy, meaning -1 oxygen. Okay? That's the only difference between the two sugars. Ribose versus deoxyribose. Deoxyribose is just missing 1 oxygen. Okay, so let's compare RNA and DNA a little bit more. The 3 major differences I want you to know between RNA and DNA. Firstly, RNA is single-stranded. You can see that here. RNA, it's just 1 strand, whereas DNA is that double helix, those 2 strands wrapped around each other. Our second difference is that the sugar in RNA is ribose, which we just talked about. It has that extra oxygen. And then the final difference is that RNA is going to contain the nitrogen base uracil instead of thymine. So if you look at this picture, we have DNA and we have 4 bases, C, G, A, and T are thymine. In RNA, we still have C, still have cytosine, we still have G, guanine, and we still have A, adenine. Those are the same in DNA and RNA, but what's different is that in place of thymine, we now have a different nitrogen base called a uracil. Okay, so those are 3 differences. RNA is single-stranded, it has a ribose sugar, and it will contain uracil and not thymine. And what is the main job of RNA? Ultimately, RNA is used for protein synthesis. Its main goal is to direct and facilitate the production of proteins. And there are going to be 3 different types of RNA that all work together to achieve this job. Okay, so we have 3 different types of RNA. The first is messenger RNA, and this messenger RNA will bring the information from the DNA out to the cytoplasm, and you can see a picture of that here. Then we have ribosomal RNA, which will clamp on to the mRNA out in the cytoplasm and use its information to assemble amino acids, which are the building blocks of proteins, in the correct order. So essentially, this ribosomal RNA, our ribosomes, are going to be building proteins. And they're going to do this with the help of our third type of RNA, which is transfer RNA. And the transfer RNAs are going to actually transfer or bring each amino acid to the ribosome as specified by the coded messages in the mRNA. Now we're going to talk more about these different types of RNA, specifically these 2, ribosomal and transfer RNA, more in our next video on translation, because that's when we talk about going from RNA to proteins. But for now, we are going to focus more on how we make this messenger RNA from DNA through transcription. We go from DNA to RNA through a process called transcription. And in transcription, a portion of the DNA strand will be transcribed or copied into mRNA. And you will notice when we go from DNA to mRNA, all of our thymines are going to be replaced with uracil. There's no thymine in mRNA. And we're now going to be single-stranded instead of double-stranded. And the enzyme that will transcribe DNA to RNA is called RNA polymerase. So you can see this big gray guy here is our RNA polymerase. And it's going to attach to our DNA strand, and it's going to transcribe a single strand of our DNA into mRNA. So let's look at how this works. All right, to start, RNA polymerase has to grab onto the DNA to get started, and it will grab onto the DNA at what is called a promoter region. A promoter region is just going to be a sequence of bases on our DNA. It's a section of our DNA that basically says, hey, RNA polymerase, come grab on here and start transcribing. Our promoter region is just like a start site. So at the promoter, you can see our RNA polymerase will grab on, and then it can begin to transcribe. All right, once our RNA polymerase has bound at the promoter, it will copy just one side of the DNA. And the side of the DNA that is copied, if you look down in this picture, the side of the DNA that is actually copied is going to be called the template strand. And the RNA polymerase will read that template strand and add complementary bases to build a new mRNA strand. This is just like in DNA, where we have complementary base pairs, except for now we will have uracil instead of thymine. All right, now the process of transcription, RNA will be transcribed by RNA polymerase in the nucleus, which makes sense because remember, DNA can't leave the nucleus. So we need to copy it inside the nucleus. And once we have our mRNA, that mRNA can leave out to the cytoplasm to ultimately make proteins. Okay, so let's practice transcription. This is not going to walk us through the whole process of transcription, but this little website will allow you to practice your base pairing when you transcribe DNA to RNA. So I will post the link to this little website if you want to try it on your own at home. It'll be in the Google Classroom. But for now, I just want to walk you through a little bit of an example. So if I pop into this little virtual website, you can see our double-stranded DNA here, which means we are in the nucleus right now. And what would happen in transcription is your RNA polymerase would attach to your DNA at the promoter region, open your DNA up, and begin to add complementary bases to one side of this DNA, the template strand of DNA. Okay, so let's open that DNA strand up, and just like RNA polymerase, we can add our complementary bases. Now remember, there is no thymine in mRNA, so anywhere that you would normally put a thymine, we're now going to put a uracil. So let's try it out. T here bonds with A. Now A in DNA would go with T in mRNA. A bonds to U. C is with G. A with U. A with U. C with G. T with A. G with C. A with U. G with C. A with U. T with A. T with A. G with C. And just going down this template strand of DNA, we are adding our complementary bases to make this single-stranded mRNA. And you can keep going, you can keep transcribing. So if you would like to try this on your own, I highly recommend you follow the link that will be in the Google Classroom and try this on your own. Okay? Okay, so now I want you to try to practice transcribing DNA on your own. So I've given you a DNA strand here. You have A, T, G, C, G, T, A, C, G, G, C, T, A. And I would like you to try to transcribe this DNA strand into mRNA, just like I did in that little virtual website a second ago. So go ahead, grab a pencil, paper, anything to write with, and try to transcribe this DNA strand on your own. Pause the video now and give it a shot. Okay, did you do it? Your mRNA should have the following sequence: UAC, GCA, UGC, CGA, U. All right, so remember, there's no thymine in mRNA, so wherever you would have originally put a thymine, you'll replace it with a uracil. So A is with U, T with A, G with C, C with G, and so on. If you got this, congratulations. That means you understand how base pairing works, going from DNA to mRNA. If you were confused, I would go back. I would watch the sections of the video again that talk about base pairing in DNA versus mRNA. Maybe go through, practice that little virtual practice, and then come back, try this again. If you still have questions, let us know. Okay? A couple of questions to review from this video. Question 1. The actual transcription of RNA is the function of, is it A, RNA polymerase, B, DNA polymerase, C, primase, or D, helicase? That's right. It's A, RNA polymerase transcribes RNA. Question 2. Transcription occurs A, in the cytoplasm, B, after translation, or C, in the nucleus? Yes, it is C, in the nucleus. transcription must occur in the nucleus because DNA cannot leave the nucleus. It is stuck there. Question 3. Which of the following about RNA is false? A. It is single-stranded. B. It contains the sugar deoxyribose. C. It contains uracil instead of thymine. Or D. None of the above. Yes, B. RNA does not contain deoxyribose. It contains the sugar ribose. However, RNA is single-stranded, and it does contain uracil instead of thymine, so those were true. All right, question 4. The ultimate function of RNA is to facilitate the production of proteins. Is that true or false? Absolutely true. RNA is so important because it facilitates the production of proteins. All right, so here's a quick recap. This is where we are right now. DNA has been transcribed into RNA in the nucleus. We have gone from double-stranded DNA to single-stranded RNA, and we have replaced thymine with uracil. That RNA has then been shipped out to the cytoplasm, where it's now ready to facilitate or direct the production of proteins. So we've covered DNA to RNA. Up next, we're going to talk about how those proteins are actually made. All right, and that is the end of video 2. We only have 1 to go. Please post any questions or comments in the Google Classroom so we can talk about this material. And as always, remember that Mrs. Liu and I are here to support you. So if we can help you in any way, reach out, let us know. And don't think that I forgot about the hedgehogs. As a thank you for watching, photo 1, hedgehog goes camping. Look at how cute. And a little hedgehog on a skateboard. Alrighty guys, have a good one. I'll see you next time.